welcome to our first Pharmaversity podcast from SCHOTT. In fact, this is our first recording, and it is a very special one already. In these turbulent times in April 2020, we are challenged by a virus, by social distancing, by working remotely, and by total isolation. Speaking of isolation, and that's why my guest today is not in the same room with me, I have Florence Buske in the line. She's in Paris right now, isolated, but I'm very delighted that she will answer some of my questions today. Welcome, Florence, and thank you very much to have you here.
Hello, everyone. Thank you for joining. I'm very happy to have such a big audience and to share all our exchange with you, because it's the first time since a long time we can do this. And I can tell you, I'm not alone. You might hear that there is some zoom around me. Summer is coming, and I couldn't get rid of the bee before starting this podcast.
Wow, that's great to hear that that even summer is coming in Paris. Here in Mainz, the sun is also shining today. Florence, you are a product expert for our glass primary packaging platform, which includes vials, cartridges, and ampules. We know each other quite a while now, for a few years, but I can tell you, I always learn from you new things while we speak. I'm pretty sure this will be the case today again. So Florence, you will give us more insights today on delamination and leachables, but also you will shed a bit more light on the secrets behind EVERIC Pure. Shall we jump right into the questions?
Yes, please.
So first of all, while I prepared this interview, I believe we should explain what the difference in technical terms really means. For me, as a non-expert, what exactly is the difference between delamination, leachables, and extractables? And this also covers, let's say, the whole scope of today's interview a little bit.
Yes, thank you, Björn, for this question. I think this is a question that comes up very, very often. Every time we do trainings or meet customers, this is a request. And so very important is that the extractables are compounds that can be removed from a product. In our case, we're talking the glass today, when subjected to a solvent. And this means that those extraction studies are often used to replicate the worst case scenario for our customers. And it shows how many compounds in our case for the glass could potentially become leachables. And this kind of studies are often required by the FDA or other regulatory bodies. And in general, to help the customers here, we give extractable statements because they are simple to do. And this is, for example, something we cannot provide for leachables. Why can't we provide this for leachables? For the simple reason that now we talk about the compounds that might migrate from the glass into the surrounding media, so inside our container, the drug, under intended use conditions. And intended use conditions means that we are talking from the glass, but in contact with the selected drug product. And we don't have it. So this is why we can't provide this. And you have seen today in our presentations and publications, we show leachable studies. That means that in this case, we conduct those studies with buffers and not only with a solvent like for the extractables. To go deeper, it's leachables are compounds or in our case elemental ions that migrate from the container, or if it's in a system also with the closure system under normal, as I said, in use storage conditions, and to which a patient can be exposed during the intake of the drug. So that is very important to have always this link. And so talking about the glass, what can happen or what can be triggered by the leachables, it's a shift of pH value. We know that, for example, storing water for injection or inactivation of a drug, for example, antibodies or monoclonal antibodies, aggregation of proteins, with, for example, the silicone, generation of insoluble particles, aluminium is something that people know here, or precipitations. So talking now more in the region of delamination, because you did also ask this, is we can distinguish two things. For leachables, one is an hydrogen attack. This is in the acid range below pH of 7. Here you have an ion exchange and a rise of pH. In the alkaline region with a pH higher than 7, we talk more about a hydroxide ion attack. And this attack releases network modifiers into the solution, and this attack is continuous. And so delamination is not only an interplay between two different variables comprising container properties and the chemical composition of the drug formulation, but also the filling and the storage parameters.
Okay, so if I understand you correctly now, you explained already a little bit delamination. Is there, what exactly happens with delamination or with what exactly happens with the glass or the glass surface when we use the term delamination?
So to make it short, or as short as possible, glass delamination is the result of a local corrosion. It takes place in the bottom near area, as we say, of the vial. And those glass flakes that appear detached from the upper surface layer of the glass, and it's a local depletion of sodium and boron in the zone between this wall and bottom. And because you ask me more specifically, it's those thin flakes that appear have a thickness of smaller than 500 nanometers and lateral dimensions up to 1000 micrometers. So those delamination of flakes in glass containers have become a serious quality concern in, let me say, the last 10 years. It's not that they were not known before, it's just that recalls have caused serious attention. And it's that serious because glass delamination typically occurs weeks or months after filling. And so you cannot see it right after filling, you cannot inspect it. And they are, as I indicated, a lot of factors that contribute to that. So what differs now is that you can only do for this phenomenon predictive studies, as I said, with the leachables, to give results that allow for a graduated assessment of increasing risk of delamination, and also to detect early stages of this phenomenon so that you can select the appropriate container for your formulation and proactively prevent the delamination instead of just monitoring for the presence or absence of flakes. So if you monitor only flakes, you may not see seldom outlayers that you may have with outlayer containers. And here, if you want to go deeper, you can also have a look at the comparative delamination study that has been given in 2017. It's, by the way, the most downloaded last year at the PDA journal. And this work demonstrates the capability of a container compatibility testing approach to determine the impact of very precise buffer formulations that are used quite often in the biologic segment and that we consider relevant. And here you can see that you can generate this effect of delamination and that it is a risk. So all in all, if we talk about the root cause now of the delamination, and we talk containers here today and what the influence of the container is, we have a root cause that is well known today. The principle is simple, if I can say so by explanation. It means that volatile components like the boron and the sodium that we can find evaporate from the glass bottom and form an inhomogeneous microzone in this near bottom area. So you have this delamination zone, and this delamination zone is a local zone, as I said, that is critical to phase separation. And phase separation means due to this boron and sodium insertion, you can have then later this issue that I described at the beginning as a result of local corrosion.
Wow. Okay, this is highly technical. From my understanding, like you mentioned the concerns and the critical aspects of delamination. The question rises immediately, is there 100% protection against delamination? Or maybe asked differently, under what conditions and circumstances does delamination happen? Is there any, let's say, idea that you have, or can you give, let's say, more insights on that to our listeners?
I have difficulties to say 100% protection. So what I can say is that if you want to go almost to 100%, it means that you need to protect yourself against this potential interaction with your product that you want to commercialize. And this is why you would need this predictive testing. So for this predictive testing to help in the region of going to the 100%, is that at SCHOTT we have assembled test kit with on the one hand we have so-called DELA test vials. That is vials that fulfill the hydrolytic resistance requirements according to the norms. But on the other hand, they fail what we internally call the quick test, which is a test to check if additionally a threshold is maintained that is well known for our production to have vials that are not prone to delamination. And on the other hand, we have exactly those vials that we call EVERIC pure that are resistant to delamination, and on the other hand, also fulfill next to hydrolytic resistance values the quick test showing that the vials have been produced in such a manner that we reduce as much as possible the potential risk of delamination from the container side. So this is what we do to support potential customers or customers in their studies to reduce the risk against delamination. And then the judgment, if this is 100%, is to be done by the testing with the real product at a specialized lab by our customers.
Okay. You mentioned the quick test as a release test. Is that something that you can share what exactly is done during that like executing that quick test, or is that something that you will share with directly with the customers?
No, we share with the customers, and I think it's well known. It has also been published. So what we do is normally you have with the hydrolytic resistance testing that we all do a ratio of contact surface that is very small. Why? Because we fill 90% of brimful. And then we autoclave 1 hour, 121 degrees Celsius. And the difference with the quick test is that we need to distinguish and test especially this microzone that is prone to delamination. And by the way, the quick test is not only testing the delamination, but also the low fillings then, and the leachables in, that correspond in this region of the vial. And what we do here is that we have all in all 6 hours autoclaving test that is dispatched in 2 parts. The first part is the vial is empty, head down in the autoclave for 2 hours. And that is a very corrosive effect that we achieve here, showing glass attack as a translation. And then the second step is we take this already aggressed vial and we fill, depending on the tube format, different content. In the case of a 2R vial, this is a 16 millimeter tube diameter and a height of 35. We would fill 1 ml. This is approximately 1/3 of the container. And then we do additional 4 hours autoclaving. And the result is then tested with the AAS. And we have defined different thresholds for those different fillings in the different container sizes, depending on the tube, to show exactly how good and bad the containers are.
Okay.
And
Wow.
so we can look at our production. Yeah, if we produce well and if we reduce the risk for our customers, and we then are secure that we do not produce those outliers that are so dangerous for recalls potentially in the pharma industry.
Okay. I remember in your recent presentation on EVERIC Pure that you stressed the point of the low filling volumes of most biologics are filled in a small vial, but as you just mentioned, 1/3 is only filled. How does, and you mentioned the point of leachables is very important as well to consider that point. Why is leachables such a risk, especially for the low filling volumes?
So maybe to jump a little deeper in this hydrolytic resistance testing, the pharmacopoeia method for the measuring of the chemical durability is the hydrolytic resistance test, as we say, which is, according to the USP 660, filling the vials 90% of its brimful volume. And what we know is that the innovative drugs that come now to the market, and it will be the same with the situation we have right now with the COVID. If we talk, for example, vaccines, it's not very high filling. So an increasing number of innovative drugs are filled significantly below the nominal volume. This means for a 2R vial that we just said with a height of 35 millimeters, that we are talking here about 1 ml or lower. So in the case of 90% brimful of a 2R vial, we talk 3.6 milliliter. So this is not the real case. As a consequence, the determined hydrolytic resistance is not representative of the concentrations of leached glass elements, as we have discussed before, and especially for low filling volumes, and which are the real case. And this attributes to 2 main factors that I just have touched briefly in your question before. First, an increasing ratio of wetted surface to volume, and second, an increased leaching tendency typically observed with borosilicate glass of the wall near bottom area, as described before, especially when standard manufacturing is applied. Yeah, so the standard manufacturing here is the subject. So this is why I say it's not representative, and why the leachables are a risk for the low filling volumes is because of the seal zone. And if we look at common buffers that we have, that we know today that have been used or are used for biologics, to talk about biologics, which include the vaccines and the other drugs that go to oncology or else, is that we have like 2 handfuls of buffers, and they start at a pH of 4.6 or 8 with a citrate, but most of them start only at 6 and go higher. And at the beginning, if you remember, I talked about the alkaline range higher than 7. So here we have those buffers that have pHs that are far above 7. And so the leaching effect, and especially combined now with these low fillings in the seal region, play a major role, not only for the delamination, but also for the drug container interaction. And I think for every pharma company, the stability is the most important. And so here we talk clearly stability over the shelf life.
And this probably has also something to do with patient safety, right? At the end.
Yeah, at the end of the day, I mean, we are not the pharma company, but it's all about the stability of the product being active, the product fulfilling its needs. And so it's all about patient safety, from the flake issue with the delamination to the leachables and stability of the product. We said at the beginning, shift of pHs and activation of the drug, and so on and so on. So this is all at the end related to the patient safety subject. Yeah, you're fully right.
So you already mentioned the different kinds of drug formulations which are specifically prone to leachables. You mentioned the biologics and the vaccines. Is there anything else, or does that basically cover everything that should be mentioned here in this case?
So I would like to say yes, but to be honest, I don't know. For the simple reason that today we know about water for injection, we know about the diluents and the difficulties, we know about the biologics, especially the big proteins, but even smaller proteins have higher pHs, so it's case by case that makes it so difficult. And then all those new products are completely new with new issues and new findings. So I believe that we have covered quite a number of potential issues with a product that is not produced in a standard way, but in an improved way to counterfight those potential issues, like the EVERIC Pure. But I don't know what comes tomorrow with some and other stuff. Yeah, we today at the beginning of understanding what this would mean. So we might hear each other again for answering those questions in the future.
Great. I remember, because you just mentioned EVERIC Pure. I remember the chart offered a Vial DC, or it is called a Vial, which is called Vial DC, delamination control. Is there now a little difference between Vials DC and the current offering of EVERIC Pure that you mentioned also in your presentation?
Well, you know, I'm very fortunate. I started in the pharma business in almost challenging times. So that was when this delamination story grew big, and I'm here. You can hear the bee, and you can hear the and you can see that. What have we done? We first looked at our core competence, and our core competence is converting vials in this case of delamination. And so we looked, what can we do from the converting side? And the result from the converting side, and a patented process, and also the testing with a quick test that also is patented. This is something that has been improved on the converting side, and the result was the shot vials DC. But then over the time we understood that we cannot look at it only from our very singular converting path. We have to include also the glass that we take. So we have worked with our tubing suppliers, and we today use the Violax CHR, so that's controlled hydrolytic resistance. They do a threshold also on their tubing product to fulfill the best possible hydrolytic resistance value, so that we have even further improved and stabilized the shot vials DC. And as it was not a real brand name, we said, following, and you know it best, to call it EVERIC Pure. So part of the EVERIC Pure is the shot vials DC historically.
Okay. And I remember from one of my first learnings that you gave me was EVERIC Pure is just one feature of or a module that can be, let's say, individualized by our customers. So it's specific. Are there any other modules or features that can be combined with EVERIC Pure? Can you explain that a little bit? If yes.
We have the perception at SHOT that our customers want to pay only for the needs they have, not for something that they don't need. So if I offer a one-fits-all solution in the packaging, then they would have to pay for an extensive number of things that they don't need. And that's why we chose a modular concept, which is the EVERIC Pure, that we have just talked about. And you're fully right, we have added modular packages that can be added or also that can be purchased separately. So the second module is the EVERIC Strong. Here it's all about glass strength. So it's not a chemical resistance subject, it's more a machinability subject in cases you need higher resistance in your process for the glass. And then a third package that can also be added to the first two, that's the EVERIC Smooth. The EVERIC Smooth is an outer coating that can be added to maintain the strong module or package over your own process. So not everybody is buying ready to use. So they have glass to glass contact, glass to metal contact, they have sticking, climbing, whatever is the issue. And so this is an additional package. And I can already tell you that the family is not yet complete. We are somehow pregnant.
Nice. That's good to hear. So you are already working on, let's say, extending the family with new children.
Right, which is the aim of a family, huh?
That's right. Sounds very promising. And this leads me to my last question, which is probably the hardest one. But obviously, once in a while, the question is raised on how leachables and delamination work with other glass types in the pharmaceutical primary packaging container area. Do you have any thoughts on that, or is that something that needs to be recognized as well, or do you want to share that with our listeners?
Yeah, well, it's a difficult subject because in the stopper area, different recipes, bromobutyl or chlorobutyl have been discussed for a while. With glass, it has been always distinctive in type 1A or type 1B, and now comes new glasses. So it's up to our customers to see how they will work with this. What I, if I'm asked like you do, tend to say is that there is from our tubing supplier in 2017 a nice PDA letter, and those results of a study have shown that 60% of the total amount of extractables of the borosilicate was lower than in comparison to, for example, to glasses that are discussed today, like aluminosilicate glasses. Additionally, I don't like this question so much, to be honest, for the simple reason that if we talk glasses, the answer of this tubing lady is the right answer. But if we talk now the container, we know that for aluminosilicate containers, there's a complex chemical treatment of the inner surface. And this brings or will bring new phenomenons that we are not able to assess today, even as a glass company well known as SHOT. And this means for pharma companies that listen to us, from my point of view, that today's expectations or expectation for tomorrow's drugs would need to be reevaluated if you go the path of a new glass. Yeah? So this is my personal point of view on this.
Wow, thank you, Florence, very much for answering all these questions from a non-expert by an expert. I can tell you already, although you are in Paris and I'm in Mainz, I felt pretty close to you. I learned again.
Same with me.
I learned again a little bit more on delamination and leachables, and I'm already looking forward to our next interview. I'm pretty sure we have more insights to share with our audience, and I'm very happy that you were the pilot for this first podcast, and I'm looking forward to do it again. Thank you very much.
Thank you, Björn, so much. I'm looking forward also to drink a coffee with you again and see the customers that are now listening to us only. It's quite strange times. Now I will take care of my little bee and give it freedom again. And I thank you so much for today. It was my pleasure. Also seeing everybody. That's quite strange in those times. So take care all over the globe. Thank you very much.
Thank you, Florence. Goodbye.